Hi everyone, we're going to talk about DNA replication and cell division with this little mini lecture. In a second, we're going to take a look at this video that's on YouTube and it's titled Live Mitosis. As you watch this video, I want you to think about the following questions. The first is, what is happening? The second is, what is moving? The third is, why are things being rearranged? And by that, I mean the things that are moving. Why are they being moved around? The fourth question is, when does this happen? And the fifth question is, where is this happening? So let's take a look at this video. You can see that there's a clump of sort of longer Cheeto-shaped things, and they're beginning to move around in the center of your video. You can kind of see they're sort of down the center of that circle area. And they look even more lined up now. You should be able to see that that line was pulled apart and that now you have two separate areas that look very similar to what you started with in the beginning. So let's talk about what was happening in this video. I'm going to play the video again while I talk to you about it. In this video, you are actually able to see mitosis happening, and mitosis is just a fancy way to say that we have cell division of our body cells, and these are all of our cells except for a couple. So what's happening is a cell is dividing, and we have some replication of our chromosomes, which is actually what's moving. So what is moving are the chromosomes. And the reason they're moving, the why things are being rearranged, is because DNA is being replicated. We're making some copies of DNA and therefore making copies of those chromosomes. And we are moving them from one side of the cell to the next part of the cell in order to make sure that there's going to be genetic information on both sides of the cell and eventually in those two new cells. This happens anytime you need a new cell. If you are injured, if cells are older and they die, if this goes out of control, you can actually end up with cancer. But most of the time, this does this to repair and to fix cells in your body. Cell division happens in almost every body cell. It officially happens, the movement of the chromosomes at least happens in the nucleus. But this happens in every single body cell, except the cells that, these are our exceptions at least. We have sex cells like sperm and egg, so that they would not do mitosis. They do another process that we'll talk about later. And some of your nerve cells, so some of the cells found in your brain and some of them found in your spinal cord would not do this. That being said, that there is actually current research saying that some brain cells do this, and there's stem cell research that is currently being used to teach cells that don't naturally do mitosis to do mitosis to repair paralysis in patients that have had a spinal injury. So let's continue on. The reason we're talking about this, remember, the reason I showed you that video is that we want to look at how structure of DNA is connected to its function. So how is something's shape and its form, how is it connected to its function? And that means what it does. Here are the 4 basic steps of DNA replication. The first thing you need to know is that we start with our original DNA in your worksheet on replication. You guys read that it was called parental or parent DNA on occasion. That doesn't they're not referring to the fact that you got that from your parents. What they're saying is the original, or the DNA you start with in cell replication and in DNA replication, is the DNA that was originally there. So just one DNA molecule. Remember, a DNA molecule has two strands and they're connected together through base pairing. The next step is going to be to unzip and unwind DNA. And then again, we have base pairing, and we have, in the end, our goal is to have 2 copies of the original DNA. So let's take a look at this. We're going to start at the top, and then we're going to move our way down the pictures. So at the top, you can see a really simplified DNA molecule. It's only showing four bases on each side. But you do have a DNA molecule that is, if we were to look at more of it, it would be a double helix. It has both sides. You can notice that they use the base pairing rules. G's and C's are together and A's and T's are together. That's your original DNA molecule. The second step is going to be to unzip and to unwind. There's a enzyme, which is just a fancy protein, that is going to unwind and unzip the strands of DNA, so it's going to separate that. The enzyme is called helicase. You don't need to remember that. If you're taking AP Bio, you will have to. But there's an enzyme, a molecule, a protein, that is going to cause DNA to be unwound and untwisted, unzipped. The third step, so the first one we see that has the yellow bases in there, that one's showing that there's another enzyme that's going to come in where we have our original DNA separated. The original is kind of the purpley blue color. And that original DNA is going to be the template for the new strands of DNA that are going to come in to make 2 new DNA molecules. So what happens in this is DNA polymerase, the name of the enzyme. Remember, you can just know that it's a protein that comes in, and it's going to read DNA. So at the left on that third picture down from the top, you see a G on the original DNA strand, and the new base that's coming in, that's being brought in by this enzyme, is C. And then you can see it goes TAC, and so it's base paired. T and A go together, A and T, and C and G. And again, you can see that the base pairing is also done correctly on the right-hand side. We're making a new complementary strand of DNA based off of the original strand. So the new one is the yellow, and the purpley blue one is the original. Now let's take a look at the very bottom one. What I want you to know from the very bottom picture is that now we have, at the end of DNA replication, we have 2 new DNA molecules that are formed. They are exactly identical to each other, and they are exactly identical to the original. So if you were to compare the bottom 2 to the very top one, so take a look. The first base pair is GC. You can see that on both of the bottom ones. The second one is TA. You can see that next. The next ones are AT and C and G. So at the very end, we have 2 new, sometimes called daughter DNA molecules. They're exactly identical, and we've now replicated DNA. We've gone from 1 DNA molecule to 2. This type of replication is called semi-conservative replication. So again, you have a picture at the right, and all that means is that each new copy of DNA has 1 original strand. So in your picture, you have a blue strand that is the original DNA molecule, and you have 1 new strand of DNA that was made during the process. And they use like a pink purpley color in here to represent the new one. Each new DNA molecule during replication will have 1 new and 1 old, or 1 original strand of DNA. And this is to prevent any mistakes or any errors. This is to keep our body working correctly.